In this video, we're going to be talking about DNA supercoiling. And the supercoiling of DNA allows that DNA to become packaged much more tightly. And that's what's necessary to fit DNA into the nucleus of a cell. In addition, as you'll see, the packaging of DNA is also a way to regulate gene expression. And this becomes an important part of the journal article that you're reading. Now, this is what supercoiling is. Here we have double-stranded DNA, right? The two strands twist around each other. And if we don't do anything, if we allow DNA to remain in its relaxed state, it just forms, if we're talking about a plasmid, a circular piece of DNA, it just stays flat. But if we either overwind this DNA or, more commonly, underwind this DNA, what happens is that there now becomes a tension, and to relieve that tension, DNA forms these supercoils, where that axis, that imaginary axis, folds over on top of itself. Very similar to what you would see with these old-fashioned telephone cords. And you'll notice that when DNA is supercoiled, it takes up much less area than when it is not supercoiled. And so when you look at DNA in most cells, it does not exist in its relaxed state. Now, the relaxed state, let's take this example here. The relaxed state of this portion of DNA would be a state in which there are 8, right? One strand wraps around the other 8 times. And so we say the linking number here, the actual linking number, 8, is equal to LK naught. Now, LK naught is the linking number of relaxed DNA, of a particular stretch of relaxed DNA. And we'll talk more about linking number in a second. Now, if you were to then unwind the 2 strands of DNA, right, to basically take one end of that DNA and twist it so you're unwinding it. So now instead of having 8 turns, we now have 7 turns. That means our LK, our actual linking number, is going to be less than our linking number of the relaxed DNA. And so that causes strain. Okay, that's gonna, when we have an LK that is less or more than the LK naught, that's gonna cause a strain. Now, we're specifically talking about a situation in which cells are underwound. The vast majority of DNA in cells is underwound. Okay, and that underwinding leads to strain. Now, what can happen then with that strain is either it forms a supercoil to relieve that strain, and that's the most common. But the cell can use this strain because when the cells remove that supercoil, when they kind of force it open, okay, not changing the LK or anything like that, when they force it open, what that does is it forces strand separation, and that strand separation is important for all kinds of biochemical processes such as DNA replication and transcription. So this underwinding of DNA allows 2 important features. It allows for supercoiling and the compaction of DNA, and it allows for easier strand separation, which allows for more easily for transcription and DNA replication. Let's define some of these terms that I've sort of tossed out there. Now, linking number is if we take a look at a molecule of DNA that is topologically constrained, meaning that we cannot twist it anymore, right? It's linked to the other strand of DNA. Now, we can do this with a plasmid because the 2 ends are connected to each other, so there are no ends. But we can do it with linear chromosomes as well by attaching proteins to the 2 ends of the chromosomes so the 2 strands can't unwind from each other. And when you see this, when you have a topologically constrained 2 strands of DNA, then what you do is you count the number of times one strand wraps around the other strand, and that is the linking number. So the linking number here is 1, and the linking number here is 6, and the linking number here is about 30. Now, the inherent structure of DNA is such that the most relaxed state of that DNA, the state at which it will not supercoil, is when the linking number, and we call that LK naught, is equal to the number of base pairs within that DNA divided by 10.5. Because if you look at the structure of double-stranded DNA, if you look at the structure of helical DNA, right, double helix, that term, helix, and you look at the bases, and it's about every 10.5 bases where you make one 360-degree turn of one strand around another. And so that's the most relaxed state of the DNA, the state at which it will not form a supercoil. And that we refer to as LK naught. Now, the only way that you can change the linking number, okay, of a particular topologically constrained molecule of DNA, the only way you can do that is by literally breaking the covalent bonds that connect the nucleotides. Basically, if you break one of the chains of DNA, that's the only way you can do that. You break one of the chains, and then you unwrap it a little bit, and then you link the chain up again. Okay, that's the only way to change linking number. Now, what you can change without breaking DNA, without changing the linking number, is you can change the amount of times that you get one strand wrapped around another to cause writhing. Okay, so twist, a little confusing, is similar to linking number, but you can kind of change the twist by changing the writhe. Now, twist is loosely defined as the number of turns of DNA, and I know that sounds a lot like the linking number, but it's subtly different because you can change the twist without changing the linking number. But when you do that, what you're also doing is you're changing the writhe, you're changing how much that DNA is in supercoils. But here's the thing. The twist is always going to try to equal LK naught. The twist, that DNA is always going to, regardless of what it does to the writhe, it's always going to achieve or try to achieve the number of twists divided, the number of base pairs divided by 10.5. It's going to try to equal that LK naught. Let's take a look at what I mean by that. Here we have relaxed DNA. Now these numbers, I got this from a textbook in which they used LK naught being the number of base pairs divided by 10. So it's not 10.5. So just bear with me with regards to that. Now the number of base pairs in this circular DNA, in this plasmid, is 360. And so what that means is the LK, the actual LK here, is going to be 360 divided by 10. So the linking number is 36. And the twist here is always going to try to be the same as LK naught. Well, and here LK naught, the number of base pairs divided by 10. So the twist is also 36. So our twist is equal to LK naught. Our linking number also happens to be at LK naught. So linking number equals twist plus writhe. If the linking number is 36 and the twist is 36, that means our writhe must be 0, and indeed it is, which means we don't have any supercoil. But if we take this plasmid and we incubate it with a topoisomerase, and we'll talk about topoisomerases in just a minute. Topoisomerases are enzymes that cut double-stranded DNA, either cut one strand, or there are topoisomerases that cut both strands, and then they pass those strands through each other, thereby underwinding the DNA. Right? So here now, with the action of this topoisomerase, we've cut the DNA, we've changed the linking number such that we've gone from 36 to 32. But remember, regardless of what happens to that linking number, the twist always tries to equal LK naught. Always tries to equal the number of base pairs divided by 10.5, in this problem, divided by 10. So that twist, because we haven't changed the number of base pairs in our example here, so the twist is still going to stay at 36. But now we have this equation. The linking number is 32 equals the twist at 36. Well, that doesn't equal each other, so we have to toss in writhe there. And the writhe must be -4. So now 36 minus 4, excuse me, minus 4, not 34, minus 4. 36 minus 4 is 32. Now that works. And so this is what would happen to that DNA that has become underwound because of the action of a topoisomerase. Now, what you can do here, though, what the cell does is it uses this as a storage of energy, which means that if you undo these writhe, if the cell undoes this writhe, so you're back at 0 now, what that's going to force is the DNA to untwist in a particular region. And when that DNA untwists in that region, right, go from 36 to 32, the two strands of DNA separate, and that can be used by the cell to do things like transcription and DNA replication. So in that way, these negative supercoils that form as a result of underwinding, make it easier for the cell to separate the two strands of DNA. Now, almost all cells of all organisms, the DNA is underwound, leading to negative supercoils. But there are some organisms that instead of the DNA being underwound, it's overwound, leading to positive supercoils. Think about what sort of organism might need that DNA to be overwound to have positive supercoils. I'm not going to answer that, but I do want you to think about it. Let's do a sample problem. And so what I'm asking you here is that we have a 1050 base pair circular plasmid, and that plasmid has a linking number of 94. And what I want you to tell me is I want you to tell me how many supercoils are in the most stable form of this plasmid. So essentially, and what form of supercoils are these? Are they positive? Are they negative? So let's do this problem. The first is we have to come back to our little easy equation. The linking number is equal to the twist plus the writhe. And then we got to figure out, well, what's 94, right? We're given something right now. We're given, the linking number of 94. So we can put that in there. So now we know 94 is equal to the twist plus the writhe. Okay, but we need to still fill stuff in here. Well, what else do we know? Remember, twist will always try to be the LK naught. And the LK naught, right, is equal to the number of base pairs divided by 10.5. So let's do that. So we know that the twist is going to be equal to 1050 divided by 10.5 equals 100. Aha! So let's fill in that equation further. We have 94 equals 100 plus writhe. Okay, beautiful. So now we can figure out what writhe is because, right, that's just a simple little algebraic expression here. That means 94, the linking number, equals 100, the twist, minus 6, the writhe. So how many supercoils are in the most stable form of this plasmid? 6. What are the form of those supercoils? Are they positive or negative? They are -6. So this is how you would solve these problems. Find out what information they give you. Remember a couple things like twist equals LK naught, and then you can solve the problem. Now, one important feature here is notice that you get a particular twist, right? Or LK naught is the number of base pairs divided by 10.5. Well, if we're comparing two genomes to each other, and we're trying to compare just how many supercoils they have, you can't just talk about raw writhe. Oh, this one has 10 writhe, this one has 100 writhe, because we have to take into account how many base pairs there are in the DNA. So instead of talking just about comparing writhe, we want to compare superhelical density. So essentially, the amount of writhe per base pair. And the way you get that, the way you get superhelical density is you take the writhe of that divided by the twist. Okay? And if you get negative, if you have a negative superhelical density, that means that the DNA is negatively supercoiled, underwound. If it's a positive density, that means the DNA is positively supercoiled and therefore positively wound. Right? DNA from bacteria and eukaryotes has a superhelical density of about minus 0.06. It's that similar amongst all of these various organisms. So this negative supercoiling, this underwound DNA, has been selected for through evolution, everything from simple as bacteria to as complex as a multicellular organism like ourselves. There are two interconvertible forms of supercoil, and they both alleviate the strain that occurs when you underwind DNA. But there are two ways that can be interconverted from one another. One is the plectonemic, where they kind of make this little helical structure. And the other one, the one that's going to be very important for our cells and for the paper that you guys are reading, is something called solenoidal supercoils, where it's like the DNA is wrapped around a barrel. And we'll come back to that a little bit later. Let's talk a little bit about topoisomerases. And now these topoisomerases, what they do is that they can relax supercoiled DNA by changing the linking number. Now there's an exception. DNA gyrase in bacteria can actually add supercoils. They can change the linking number to actually add supercoils, but all the other topoisomerases within our bodies, within our cells, they relax supercoiled DNA by changing the linking number. Now, type 1 topoisomerases do this by changing the linking number by 1 with each round. And what they do is they cut one of the two strands of DNA, they pass the other strand through, and then they seal it back up. They nick it, they pass one strand through the other, and they seal it back up. That changes the linking number by 1, and this will therefore alleviate some of the supercoiling. Now, type 2 topoisomerases, they work by, let's see, we're going to start right here, and then they double-stranded cut right there of DNA. They pass the other two strands through, and then they seal things back up. Right? Now, those type of topoisomerases, number one, they're going to require ATP, where the type 1 topoisomerases didn't require ATP. And then in addition, they're going to relax DNA by linking number by 2 each time. If we take a look at a series of plasmids here, circular DNA, what we can see is that depending on how supercoiled it is, it has different properties, right? Here is one that is completely unsupercoiled, completely relaxed there. And yet we can compare it to something that's really supercoiled, and it's really tightly supercoiled there. But yet, each one of these is the exact same molecule that just exists as different topoisomers, identical sequences of DNA that differ in the degree of supercoiling. If you were to put this on gel electrophoresis, you would be able to separate each one of these topoisomers from each other. And we'll look more into that sort of experiment when we talk about DNA electrophoresis a little bit later in the course. Now, topoisomerases are necessary constantly for the proper function of many of these biochemical processes that must occur to DNA. For example, if we have an RNA polymerase that is transcribing a stretch of DNA, what it has to do is it has to unwind the two strands of DNA. And as you force those two strands to unwind, what ends up happening is that DNA ahead of that replication fork begins to overwind, begins to form these positive supercoils. And if this is allowed to continue, those overwound DNA would get to so extreme that the DNA polymerase would no longer be able to separate the two strands of DNA, and DNA replication and transcription, as shown here, would stop. And so what we have is, as DNA replication is occurring, as transcription in this particular example is occurring, there are topoisomerases that work to alleviate this overwound DNA and these supercoils. So they're necessary, constantly necessary for these type of processes. All right, so the final lecture that we'll have for this particular meeting is with regards to chromatin structure. And this is going to be important for understanding the paper that you're reading as well, and we'll make this another video.